Polyadenylation is the process of adding a long chain of adenine nucleotides to the end of an RNA molecule. The name itself explains the process, so polyadenylation literally means the addition of many adenine bases. This added chain is called the poly-A tail. Now, this process mainly happens in eukaryotes during mRNA processing. In eukaryotic cells, the RNA formed directly from transcription is called pre-mRNA. But this pre-mRNA is not ready yet to make proteins. It first goes through processing steps like 5' capping, splicing, and polyadenylation, which happens at the 3' end of the pre-mRNA. First, RNA polymerase transcribes DNA and produces pre-mRNA. As transcription continues, a specific sequence appears near the 3' end of the RNA. That sequence is usually AAU-AAA. This is called the polyadenylation signal sequence. This sequence is extremely important because it tells the cell where polyadenylation should occur. The RNA is usually cut about 10 to 30 nucleotides downstream of the AAU-AAA signal. Remember that this is just a signal which helps recruit special proteins that prepare the RNA for cleavage and poly-A tail addition. One major protein complex called CPSF, which stands for Cleavage and Polyadenylation Specificity Factor, binds to the AAU-AAA sequence. Another protein called CSTF binds to a GU-rich region located slightly downstream. Together, these proteins position the RNA correctly for cleavage. Then the pre-mRNA is cut. After cleavage, the newly created 3' end becomes the site where the poly-A tail will be added. Now, an enzyme called poly-A polymerase begins adding adenine nucleotides one by one. Interestingly, this enzyme does not need a template. During transcription, RNA polymerase uses DNA as a template to decide which nucleotide to add. But poly-A polymerase simply keeps adding adenine bases repeatedly. So the tail becomes something like this. In humans, the poly-A tail is usually around 200 adenine nucleotides long. As the tail grows, proteins called poly-A binding proteins attach to it. These proteins help stabilize the tail and protect the mRNA. Now the question is, why does the cell add this tail at all? The first function is protection. RNA molecules are naturally unstable inside the cell because enzymes can break them down. The poly-A tail acts like a protective buffer. A simple example is the plastic cap at the end of a shoelace. That cap prevents the shoelace from fraying. Similarly, the poly-A tail protects important coding regions of the mRNA from being degraded too quickly. Second, the poly-A tail helps export the mature mRNA from the nucleus into the cytoplasm. Protein synthesis happens in the cytoplasm, so the mRNA must leave the nucleus first. Another important point is that the length of the poly-A tail can affect mRNA lifespan. Over time, the tail slowly becomes shorter. This process is called deadenylation. When the tail becomes too short, the mRNA becomes unstable and is eventually degraded. So the poly-A tail also helps regulate how long an mRNA molecule survives inside the cell. So overall, polyadenylation is a post-transcriptional modification mainly seen in eukaryotes, where a long chain of adenine nucleotides is added to the 3' end of pre-mRNA.